I'm Miss Conquest, and I am a biology teacher here at Mainland, just down the hall from you guys. And I know that you're probably wondering what is going on. Well, I just want to tell you Miss Ishii is getting better. She will be back as soon as she can. But in the meantime, this is probably the best way for you guys to learn the unit. You will have to take the DIA before Christmas break. So we're going to make sure that you guys are on track and doing everything you need to do. So this lesson is going to be on DNA transcription and translation, meaning how DNA replicates and then turns into messenger RNA, which then helps the cell make proteins. Sounds like a lot, but I promise it'll make sense. Before we start, let's review DNA. All right, so we know, and hopefully already you guys know, that DNA is the genetic information that makes up all living things. All right. I'm going to give you guys just a second to pause here and answer these two questions. One, explain why the genetic code in DNA is universal. And in your own words, describe the structure and function of DNA. Go. All right, very good. So just some review. All living things have DNA. DNA is found in the nucleus, and it contains all of our genetic information. All right, DNA's discovery, I'm going to breeze through this. So 100 years ago, approximately, Frederick Griffith discovered it, and Watson and Crick are the two guys who discovered the shape being a double helix, which is like a spiral staircase. All right, two more questions. Stop, think, and review. So take a moment. What did Griffith discover DNA contained? And number two, why do you think DNA's discovery was so important? All right, very nice. So moving on, we know DNA is a double-stranded or double helix, basically a chemical called nucleic acid. So there's two sides. These two sides connect together with specific base pair rules, which you guys should be familiar with. Now, what makes up DNA? Remember the term, the nucleic acid and nucleotides. So DNA as a whole is a nucleic acid made up of several smaller nucleotides. Each nucleotide has a sugar, a phosphate, and a nitrogen base. Okay, here's just a smaller picture of the nucleotide. You have the phosphate, the base, and your sugar. All right, something I tell my classes to remember the base pair rules is a funny saying called apples in a tree, cars in a garage, meaning A always pairs with T, apples in a tree, C always pairs with G, cars in a garage. Okay, now if you guys know this already, you are ahead, and really that's all that you're expected to know right now. Know that the order of the bases is unique, so I have a different order than you do. But all living things do have the same four bases. All right, I want you guys to stop and practice. So real quick, you're going to just pair up the correct nitrogen base. Remember, apples in a tree, cars in a garage. Very good. All right. DNA is said to be the universal code because all living things have it, meaning I have the same four bases as a giraffe. We all have A, T, C, and G. So it's said to be universal. That's a key thing to remember. All right, now DNA has to replicate or make a copy of itself. If you guys go back to the last unit, you should know what phase DNA replication takes place. So this is your next question. Fill in your box. Alrighty, DNA replicates. So DNA has to make a copy of itself in order to turn into messenger RNA. Now, before we get to these steps, I want to go and kind of just run through the cell with you. So I'm going to now share the whiteboard. And if you have paper, I would recommend drawing this as well. All right. So I'm going to use my fingers. Here we have a cell. Okay, this particular cell, we're going to say it's animal cell. So animal cells have a nucleus. Remember, they are eukaryotes. They do have a nucleus. All right, inside the nucleus is where DNA is stored. Oh, that looks terrible. Let's try it again. Nucleus. Why is it doing that? Let's fix this. All right, there we go. I'm going to erase this booger. And we will move this over. All right, DNA is inside the nucleus. DNA, believe it or not, cannot leave the nucleus. And you might be wondering why. And the answer is it's too big. So if you remember, DNA is a double helix. Okay, so on the outside of the nucleus are these nuclear pores, and DNA is too big to be able to go through it. Now, if you remember, what organelle inside of a cell makes proteins? Good, ribosomes. So I'm going to draw a ribosome, not drawn to scale because ribosomes are small. But if you remember, let's go back here, make this one a little smaller. And add a new one. So I'm going to call this ribosome. Ribosomes are responsible for making proteins. But here's the catcher. Ribosomes have to get instructions on how to make proteins. And the only thing inside the cell that has those instructions is the DNA. But DNA can't leave the nucleus. So somehow the instructions have to get to the ribosome. Well, in order for that to happen, DNA has to turn into RNA. Very good. So now we're going to have RNA. So DNA and RNA. All right. DNA has to turn into RNA. RNA is a single strand, so it's smaller than DNA. DNA kind of has this double helix, right? And then you have your bases connecting. Well, RNA looks like a single strand, and it has the bases coming off just like that. So it's smaller. So long story short, DNA turns the message or the secret recipe of what proteins to make into RNA. So now RNA takes the message or the recipe to the ribosome. Now, because this RNA is carrying the message or the recipe, it's often referred to as the messenger RNA. So let me write that here. Messenger RNA. So messenger RNA carries the message to the ribosome. Now the ribosome has the information or the instructions on what proteins to make. And that is, in simple terms, how DNA instructs the cell on making proteins. We call this protein synthesis. All right. Now, let's go over some rules again. So remember DNA. If DNA is making a copy of itself, we have apples in a tree, and then we have cars in a garage, so ATCG. When DNA turns to RNA, any kind of RNA, the only kind we've talked about is messenger, but there are other kinds. So anything RNA, there is no thymine, which is what T stands for. So the base that replaces thymine is called uracil. I'm going to spell it out, URACIL. There is still C's and G's. Okay, so what does that mean? Remember on Friday you guys practiced the DNA complementary strands? All right, so basically what you did is you took a strand of DNA and you turned it into complementary DNA. All right, let's see if I can move this. For example, if this is DNA, I'm going to do up some DNA sequence, ATCG or AG, ATCG. All right, for me to make a complementary DNA strand, remember A turns to T, T turns to A, C turns to G. T, C, T, A, G. All right, now, so this second piece right here, that is called complementary DNA. So I just took an original sequence and made a copy. Now this copy, I have to turn into RNA, specifically messenger RNA. All right, so let me show you what I mean. So my DNA complementary strand is TAG, TCT, AGC. And I want to make RNA. All right, here's where it gets a little tricky. There is still adenine in RNA, so A turns to T, or T turns to A. However, instead of the A turning to a T, it's going to go to a U. And then the G's and C's stay the same, so they're never going to change. So now we have TCT, so I'm going to have AGA. And now I have AGC, which will go to UCG. All right, so we just made a RNA out of our DNA strand. Okay. All right, you will practice this in just a second, but now I have to show you how this RNA has the codes, we call them codons, to make the proteins. All right, if you recall, proteins are long chains of amino acids. So if I were to, yeah, that's too big, I don't like that. Undo. How do I undo? Just draw. All right, if I have a bunch of amino acids, these are the building blocks of what makes up a protein. Okay, remember amino acids are the building blocks of proteins. Each one of these, let me change the color here, is going to give me a specific code for each amino acid. So there's a code here, a code, code. All right, we call these specifically codons. C-O-D-O-N. A codon is 3 letters of an RNA sequence that codes for an amino acid. So in this particular RNA strand, I'm going to have 3 codons. Okay, I have the AUC, the AGA, UCG. Now, how do I come up with the name of this amino acid? I'm going to introduce you to a chart that we use. Okay, and if I can pull it up, I'll be happy to show you guys. Let me see if I can now share it. You're going to have one of these at your desk. All right, so here you can see it looks like a crazy chart, kind of like a sun. And this is going to be how we uncode the name of our amino acids. All right, so on the whiteboard that I was just sharing with you, I'm going to go back to it. We're going to start with our first mRNA codon. All right, which is AUC. So if I look on this chart, you have this in front of you. I'm going to start in the middle, okay? So AUC, I'm going to write these down and then I'll go back to my screen. All right, so I just wrote down my 3 codons, AUC, AGA, UCG. All right, so let me go back here. And the first codon is AUC. So when you look on here, I'll draw it for you. We're going to start in the middle. So my first letter is A. My second letter is U. So from here, I'm going to work outwardly. So the U is right here. So AU, the last letter is C. C is right here. So I'm kind of drawing this out, and it brings me to the name of a specific amino acid. This one is called isoleucine. You do not have to remember the names of all of these because you will have this chart on your test, I promise. Down below, you see there's another chart. It's the same amino acids. It just gives you the abbreviation. So isoleucine is right here. So instead of writing it out, if I wanted to abbreviate, I could just write ILE. So these are just abbreviations. All right, I want you guys to practice the next 2 codons. It should be AGA and UCG. All right, now that you've done that, your 2 codons should be AGA, which would come AGA, arginine. So if you got arginine, you did it right. And UCG. CG. So we have serine. So if you had serine, then you got yours correct. All right. This is how the RNA codes for amino acids. Okay, so now I'm building a protein because remember, amino acids are the building blocks of proteins. All right, hopefully it makes sense. We're going to practice some more. I'm going to put up a strand here of messenger RNA. Actually, I'm going to put up DNA. And then I want you to translate it to messenger RNA. And then I want you to find the amino acids. All right, so my DNA strand. I'm just making this up. make it wider. So first question I want you to think about is how many codons are in this particular DNA sequence. Now, instead of having you turn it into a complementary strand of DNA, we're going to go straight to messenger RNA. Okay, so we're going to pretend like this DNA already copied itself. So I want you to take a second and fill out your mRNA sequence. All right, so hopefully these are the 4 that you got. And now you're going to find the amino acids. So look on the chart that you have. I want you guys to uncode each of these. Okay, so 4 codons total. 1, 2, 3, 4. All right, you go ahead and do it, and I'm going to practice along with you. All right, guys, so UAC. UAC should have coded for tyrosine. Okay, the next one, can't see what I wrote here. GGU. GGU is glycine. Just got my capitals mixed up. AUC. AUC, isoleucine. And the last one is CAA. CAA is glutamine. It's a good one. All right. Notice on your chart, the one you're looking at to uncode it, some of them are called stop. What that means is if that is the last amino acid in the chain that it's making, that's the last one. It's like the caboose. So it's a stop codon and it tells the protein that it's done being made. All right. So this is in simple terms, protein synthesis. All right. What I want you to do is work on the worksheet that you guys have. Tomorrow we'll go more into specifics about this, and then hopefully by the end of the week, you guys will be rock stars and ready to go. All right, if you have any questions, comments, or concerns, you can email me. My email should be on the top of Miss Issue's whiteboard, but it is erconque@volusia.k12.fl.us. I am just down the hall in room 312. Don't hesitate to ask or come by if you have questions. All right, you guys, I hope this helps you. Have a great day.